So now today we'll be discussing about the other topic, that is your muscles and adipose tissue. Now what you'll be able to learn is to describe muscles and adipose tissue, to describe various types of muscle and adipose tissue, to learn the histology of muscles and adipose tissue, to differentiate between different types of muscle fibers, and to differentiate between white versus brown adipose tissue. First we'll be dealing with the muscles. Now, what is the muscles? The contraction of muscle tissue is the only way in which we can interact with our surroundings and is essential to maintain the life itself. By that, it means that there is a contraction and relaxation of muscle going on in our body, and that is responsible for our movement. When we stretch our arms to pick up anything, when we flex our arms, whatever we do is because of the contraction or relaxation, or what we call it as coordination of muscles. So that is why it is said that. Now there are basically three types of muscles. Those are your skeletal muscle, cardiac muscle, and smooth muscle. Okay, we'll talk about it. Okay. Now the voluntary contraction of muscle, skeletal muscle, by that it means that when the brain sends a signal to the muscle that they have to contract. For example, I'll give you an example. For example, you touch to a very hot liquid, maybe a hot coffee, and suddenly you start feeling burning. You suddenly move your hand. That is what? That is your voluntary contraction of muscle. So what happens is the brain signals the muscle fibers that there is something hot and it is going to affect you. So you should better remove your hand. And that is what we call it as voluntary contraction of skeletal muscles. Allow us to move our limbs. Those are your, by that it means your hands or your legs, fingers and toes, to turn our head and move our eyes and to talk. Okay. So basically, voluntary and involuntary means when you do not have a control over your this thing on your movement, and voluntary means when you have a control over your movement. Now, introduction. First is, what is cardiac muscle contraction? Cardiac muscle contraction is involuntary, in contrast that of skeletal muscle. Now see, here it is the thing. Your cardiac muscle itself, it says that it is basically the muscle of your heart. Okay? Now the muscle of your heart is involuntary. By that it means we do not control the contraction and relaxation of the heart. Heart is what? Heart is basically your muscular organ that pumps blood in and out. Okay. Now we do not tell the heart to stop now, to stop the heart, or not to contract or not to relax. So this is involuntary. When the movements of the muscles are not in our hand, when they are organized in their own manner, that is called as involuntary, in contrast to that of the skeletal muscle. Although its frequency of contraction is modulated by autonomic nervous system and by hormones and neurotransmitters in the blood. Okay. Now smooth muscle. Now smooth muscle is the most diverse type of muscle. It occurs in different subtypes in different organs and is essential for involuntary physiological functions. For example, regulating blood flow, your blood pressure, aiding the digestion of food. How they aid the digestion of food? There is continuous peristaltic movement going on in your intestine that helps in the movement of your food. Moving food through the digestive system, regulating airflow, respiration, controlling the diameter of the pupil in the eye. By that what it means, that when you are trying to put a light onto the pupil of your eye, it contract and then it relax. Expelling the baby during childbirth and others. So these all are the role of the smooth muscles. Now what are the functions of muscles? Movement, that is your skeletal, cardiac and smooth muscles. Stability, it provides movement first of all, then it provides you stability. So those are through skeletal muscles. Communication, those are through skeletal muscles. Control of body opening and passages, that are through your skeletal and smooth muscles. Heat production, that is through your skeletal muscles. So basically it provides the functions in movement, in stability, in communication, in control of body opening and passages, and your heat production. What are the properties? First is excitability. That is what I've explained you. In response to chemical, mechanical, or electrical stimuli, they becomes excitability is there. Okay. And conductivity. Initiate events that lead to contraction. So these are through conduction of the signals. Contractility, that is ability to shorten substantially. So they contract, so they become shortened. Extensibility is the opposite, able to stretch. So there is contractility and there is extensibility, and there is something called it as elasticity. So ability to return to original length after stretching. That is what we call it as elasticity. Means they are able to come back to its own position after they are being told to stretch. They have the ability to come back into their own position. So there is excitability, there is conductivity, there is contractility, there is extensibility, and there is elasticity. Now there are three types of muscles as I have told you: skeletal muscle, smooth muscle, and your cardiac muscle. Now muscles, where do they arise? What are the origin? So most of the muscle tissue develops from mesoderm that give rise to mesenchymal cells. Okay, so that basically arises from the mesoderm that give rise to mesenchymal cells. Now skeletal muscles develops from paraxial mesoderm organized into myotomes in somites. You have to learn this, okay, because this is anatomy. Muscles of the head develop from mesenchyme of branchial arches. Cardiac muscles, they develop from cardiogenic mesoderm. Smooth muscles, they develop from splenic mesoderm, except of iris muscle where smooth muscle arises from neuroectoderm. So you have to remember this. Skeletal muscle, they arises from paraxial mesoderm, cardiac from cardiogenic mesoderm, smooth muscles from splenic mesoderm. What are skeletal muscles? Skeletal muscles is responsible for movement of skeleton as well as organs such as globe of the eye and the tongue. So basically we have a skeleton. So in order to move the skeleton, skeletal muscles are required. That is why they are called as skeletal muscles. Skeletal muscle is often referred to as voluntary muscles since it's capable of voluntary or conscious control. By that it means we are able to control it. For example, if we want to put our leg forward, we will do that. If we do not want to, we do not do that. If we want to stretch our arm, we'll do that. So it is called as voluntary control. The arrangement of the contractile proteins give rise to the appearance of prominent cross striations. So basically it has a cross striations in some histologic preparation, and so the name striated muscle is often applied. So it is also called as your striated muscle. Why? Because there are cross striations that you see. Now, what are the properties of skeletal muscle? So cells are long and cylindrical in shape. So if I'll show you with the help of a pointer, see these are all your striated cells. These are long. They have cells are multinucleated. So 1, 2, 3, 4, many nucleus are there. This is your muscle fibers. So cross striations, if you can see, these all are your cross striations that are present under voluntary control. What are the contractile proteins which help in contraction? So what are the proteins in these skeletal muscles which help in contraction? Those are your actin, myosin, and tenin. So basically actin, myosin, and tenin. What are regulatory proteins? Troponin and tropomyosin. So these are basically troponin and tropomyosin. Now what is sarcomere? Basically every muscle fibers have a contractile unit. So the contractile unit of skeletal muscle is sarcomere. So is the contractile unit of skeletal muscles defined as the distance between two Z disc. Now what is Z disc? I'll let you know. So for now we will focus on this. So basically how are the cells? The cells are long and cylindrical in shape. They are multinucleated. They are having cross striations. We already have discussed that they have voluntary action control. Okay. Then what are the contractile proteins? Actin, myosin, tinin. Now what are regulatory protein? Troponin and tropomyosin. Then comes is your, what is the contractile unit of skeletal muscle? That is your sarcomere. Okay. Now this is the structure. See, in this there is a highlighted part. These all are basically your the covering. Now if this is your one muscle fiber through the cut section. Now what is myofibril? It is composed of bundles of myofilaments. Okay, so they have myofibrils. Now what is endomysium? Now where is the endomysium in this? This is the endomysium. Okay. Now what is endomysium? This is an areolar connective tissue covering each muscle fiber and binding it to its neighbor. So basically, see, this is your endomysium, and it is basically covering see, these all are your muscle fibers. So they are covering all the muscle fibers and binding it to its neighbors. So see, this is one muscle fiber, another. Now they all are binded together by whom? By endomysium. Okay. Now there is something called as perimysium. Peri, as I have told you, means surrounding. So dense irregular connective tissue covering the muscle fascicle. So they are surrounded by the muscle fascicle. Okay, so this is your perimysium. Now then your what is fascicles? Fascicles are bundles of muscle fibers surrounded by perimysium. So can you see these? These all are your fascicles, muscle fascicles. These all are your muscle fascicles, and they are what? Surrounded by your perimysium. Then what do you mean by epimysium? Now covering of dense irregular connective tissue surrounding the entire muscle. That is why it is called as epimysium. They are covering the entire, not the inside portion, but the entire portion of your muscle. So you will have myofibril, then you will have your endomysium, then you will have your perimysium, fascicle, and epimysium. These all are the terms that you need to remember. See, now this is skeletal muscle structure, the whole muscle. So this is your one entire muscle, and this is your single muscle fiber. They have cut the single muscle fiber. So in this you can see that there is, now what is this? See, we'll start from here. See, this is tendon. So this is the attachment. Now this is your epimysium. Now these all are your fascicles. These all are your fascicles containing the muscle fibers. Okay. Now covering to it was perimysium. Then there is an endomysium. Okay, so all these structures are there. In this also you will be able to understand. So these all are your muscle fibers. Then this covering to it is endomysium. Outer covering to it is perimysium. Then they will have nuclei also. So all these things would be there. Now how do they organize? How do they look like? Okay. So muscles are composed of muscle fascicles, which I have already told you. Okay. They are composed of skeletal muscle fibers. And so first is what? Muscles are composed of fascicles. They are composed of skeletal muscle fibers. Muscle fibers contains what? They contains myofibrils. Okay, remember that. Myofibrils create the biggest part of sarcoplasm. They are oriented longitudinally with long axes of muscle fibers. Myofibrils are composed of thin actin filament and thick myosin filament. Now, if you remember, in previous slides, we have discussed about the contractile protein of the muscle, skeletal muscle, actin, myosin, and tinin. So this is what they are telling it here. So myofibrils create the biggest part of sarcoplasm. Now, myofibrils are arranged as thin actin filaments and thick myosin filaments. So remember that. Now, actin filaments on molecular level is composed of what? It composed of F-actin, troponin complex, and tropomyosin. So consists of troponin and tropomyosin. Now again, if you recall the previous slide, the regulatory protein for the muscles, skeletal muscles, are troponin and tropomyosin. So these are that. Okay. Now actin in the sarcomere is bound by Z line. Myosin filament is composed of 100 of myosin molecules. So basically in this slide, if you are getting confused, you just have to remember that muscles are composed of muscle fascicles. Muscle fascicles, they are basically muscle fibers. Now muscle fibers contains myofibrils. Okay. Now myofibrils consist of what? Thick thin actin filament and thick myosin filament. Okay, now we'll talk about first actin filament. What does actin filament contains? It contains F-actin, troponin, and tropomyosin. Okay, now what does myosin contains? Myosin contains myosin molecules. See this is what it will show. So this is your myofibril, myofilaments. It has your tropomyosin, troponin. So these all are your regulatory protein. Then it has your actin. This is your active site, and this is your myosin. So these are basically your myosin. So actin and myosin. And myosin contains what? Myosin molecules. Now what are the functional unit? What is the functional unit which we have discussed? That is called as sarcomere. So here we are talking about sarcomere. So sarcomere are repeated functional unit visible in the myofibrils in electron microscopy. A sarcomere is present between two Z lines. Now see this. This is your muscle, okay? Muscle fiber. It consists of your cross striations. What does it has? It has your Z disc and it has your in between H zone. So there is Z disc, H zone, Z disc. In between that, if you can see this is your I band. Then the longest part is your A band. So I, and this is your M band. Okay. Now the distance between two Z lines. So this is one Z disc and this is one Z disc. Now the distance between these two are called as your sarcomere. Remember that. So is present between two Z lines. Sarcomere is present between two Z lines. Okay. The Z line serves for attachment of actin filaments. I band is formed only by actin filaments. A band is composed of myosin filaments and partly of actin filaments. There's nothing to explain in this. You just have to remember this. Okay. Now this is your micrographic picture. So see, basically when we cut the muscles, we can either cut it into a transverse section or we can either cut it into a longitudinal section. So there is transverse section and there is longitudinal section. This is what we are talking about, your transverse section. Now what does this have? This have all these, can you see these pink areas? These all are your muscle fibers. Now inside that muscle fibers, these all blue dot that you are seeing, the pointed over here also, is basically your nucleus. Okay, and this is your epimysium. That is what we have already discussed, the covering that covers the muscle fibers. Okay, now see, this is your perimysium, which is see, what is the function of the perimysium? They cover the surface of the muscles. Okay, so this is the high power view, this is the low power view. In high power view, you can see more clearly that this single thing is your muscle fiber. This blue dot thing is basically your nucleus. So this is what it is. Now if you into the longitudinal section, then this is more cross striations. You can see the cross striations of the muscles, and these dot things are basically your nucleus. Now what is contraction of the muscles? So basically what happened is, during contraction occurs shortening of sarcomere, I band and H band. There is no change in the length of A band and actin and myosin filament. So this you have to remember. See now, if you see into this picture, let's start from one. What happened in contraction? The first thing is the myosin head hydrolyze ATP. Okay, you know what is ATP, and become reoriented and energized. Adenosine triphosphatase ATP. Okay, so myosin heads hydrolyze and become reoriented and reorganized. And myosin heads brings to actin forming cross bridges. So what they do is myosin head becomes activated and then they activate the actin also. Okay, and then it forms a cross bridges. The myosin heads rotate towards center of the sarcomere. So this is the center of the sarcomere. And as myosin bind ATP, the cross bridges detach from the actin. So there is contraction and there is relaxation. Contraction and relaxation. See, you do not have to remember this entire process. It will be too much for you. What you have to remember is in contraction, which shortening is happening and in which there is no change. So shortening of sarcomere, I band and H band, and there is no change in A band or actin and myosin filament. Now smooth muscles. So that was all about skeletal muscle. Now skeletal muscle is specialized for relatively forceful attract contraction of short duration under voluntary control. Okay, that is what skeletal muscle was. So skeletal muscle was forceful contraction of short duration under voluntary control. Now smooth muscles specialize for continuous contraction of relatively low force. So basically what happened is that in skeletal muscle there is a contraction of a very short period but of a high force. Whereas in this you see the contraction of a longer duration but of a low force. Okay, producing diffuse movements through contraction of the whole muscle mass rather than contraction of individual motor unit. What do you mean by that? By that it means what happens in skeletal muscle. For example, if we are getting burned by some hot thing, we try to remove our hand. Okay. So in this, what is happening, or best forget about this. A simple example I'll tell you. If there is a water bottle and we want to pick it up, so what we do is we stretch our arm to take the bottle and then we drink it. So there is stretching and there is contraction. There is relaxation and there is contraction. That is your voluntarily movement. But this is not happening in your legs also. You are just using one of your hand. So in this is what it is trying to show you, that unlike skeletal muscles, they produce diffuse movements. Diffuse means that are spread all over rather than to an entire area. Okay, that is specific. But diffuse means that are present over a larger area through contraction of the whole muscle mass rather than contraction of individual motor unit. Okay. Now, contractility is an inherent property of smooth muscle, which is also there in skeletal muscle, occurring independently of innervation, often in rhythmic or wave-like fashion. For example, the smooth muscle of intestinal wall undergoes continuous rhythmic contraction. That is what we call as peristalsis. I've also told you this term before. Now, in our intestine, there is continuous peristaltic movement going on, and that will help in what? Help in expelling of the food from our small intestine to our large intestine, and then expelling out of our body, the waste. So that is what is an example of smooth muscle. So now this is the micrograph of smooth muscle. Now the cells of smooth muscles are small. If you see these cells, and if you remember the cells of the skeletal muscles, these are small. Okay, and then the fibers are bound together in irregular branching. See this, see the pattern. It is bounding from this side to this side to this side. So this is called as irregular branching fasciculi. The arrangement varying considerably from one organ to another organ. So this is the micrograph. Now coming up to cardiac muscles. Cardiac muscle or myocardium exhibit many structural and functional characteristics intermediate between your skeletal and your visceral muscle. So they have the characteristic and property either of the skeletal or your visceral muscle. Like the former, its contraction are strong, like the skeletal muscle, and utilizes a great deal of energy. Now, like the visceral muscle contraction are continuous and initiated by inherent mechanism. So this is what it is. So it lies between your skeletal and your smooth muscle. So it is characterized by cross striations, intercalated disc, uninucleate cells, automaticity, composed of same contractile protein as skeletal muscle. So it has same structures like that. So this is your cardiac muscle micrograph. It is written smooth, but this is basically your cardiac muscle. So muscle scenes are contained to one or two nuclei, extensively eosinophilic cytoplasm. So you can see that this is your muscle fiber, this is your nucleus. Okay. If you can see, this will give you a continuous three-dimensional network. Now this is the basic difference between your skeletal versus smooth versus cardiac muscle. So location, skeletal muscles are generally attached to the skeletal. Smooth muscles are basically attached to your viscera, iris, blood, and cardiac muscles to your myocardium. Shape, skeletal muscles of long, cylindrical, parallel fibers. Smooth muscles, as we have seen, is short, spindle shape, and this is your branched. and blunt-ended. Striations are present, yes, in skeletal muscle, no in smooth muscle, but it is present in cardiac muscles. Number of location of nucleus, numerous, but in smooth muscle it is single. In here it is one or two central. Now T-tubules, forget about it. Sarcoplasmic reticulum, it is there. Gap junctions, all these are okay, you can read it, okay? Just for your knowledge, but these are not so much important for you right now. Now coming up to your adipose tissue, the other part. Now adipose tissue, what is adipose tissue? In general, if I tell you what is adipose tissue, it is basically your fat cells, okay? Now a specialized type of connective tissue, yes, fat, yes. Specialized type of connective tissue in which adipocytes or fat cells predominate. That is what we call it as adipose tissue, located in many areas throughout the bodies. So adipose tissue represents 15 to 20% of the body weight in men, and in women it's 20 to 25%. So basically, if we are talking about somebody who is lean, so it will have 15 to 20% of the body weight in men and 20 to 25% of body weight in women. Now, what is the main function of adipose tissue is to regulate our body temperature. So, to regulators of the body energy metabolism. What are the functions? Functions is they release hormones and a number of important factors. Okay. Second is a poor heat conductor. Then it contributes to thermal insulation of the body. So it regulates the temperature of our body. What is the other is it fills up the, not because of pregnancy and lactation, because of the normal structure of woman that is required. Secondarily, the hormones, all these things affects in difference between men and women. Metabolic rate also. So the other function is it fill up the spaces between other tissues and help to keep some organs in place. So it basically, whenever there is an empty space, adipose tissue try to fix the void. Okay. Now deposit in the form of fats and act as a shock absorber, because these all are fat cells. They are having fat inside them. They are having lipid inside them. So whenever there is a shock or a blow to the body, to some of the part, it acts as shock absorber. Okay. Now what are the types of adipose tissue? There are basically two types. There is one more type, but that is not important for you to remember. There are basically, the most common type is your white adipose tissue, the most common type. And the other one is brown. So the white adipose tissue, the more common type, is composed of cells that when completely developed contains one large central droplet of whitish yellow fat in their cytoplasm. So this is your white adipose tissue. Now what is brown adipose tissue? They contains multiple lipid droplets. So the difference is, in white adipose tissue, when they mature, they contains only single lipid droplet, whereas in the brown adipose tissue they have the multiple lipid droplet interspersed among abundant mitochondria. And because of the lipid droplets and the abundant mitochondria, they give you a darker appearance. That is why it is called as brown adipose tissue. Now coming up to the white adipose tissue. So now white adipose tissue is a specialized for long-term energy storage. So this basically their function is the long-term energy storage. White adipose cells are spherical when isolated but are polyhedral when closely packed in adipose tissue. Okay. Each cell is very large, between 50 to 150 meter in diameter, and contains one huge droplet of lipid. So they have one huge droplet of lipid that makes up to 85% of the cell weight. So the majority that it contains basically is the huge droplet of lipid, called as unilocular. Now that is important because triglycerides are stored in a single locus, seen predominantly in adult humans. Now see this. So this is there. Grossly viewed as white or yellowish mass, tissue has a rich blood supply. Both have rich blood supply, be it brown or white. Capillaries found around adjacent adipocytes meet. Numerous mast cells present. Unmyelinated nerve cells are present. What are the functions? Synthesis, storage, and mobilization of lipids. Second is your insulation. So it helps in insulating, thermal regulation, cushioning of vital organs, and secretion of hormones. Now, what is the histogenesis? Forget about what is written over there. See this. Now, this is your undifferentiated mesenchymal cells. I've already told you the difference between differentiated and differentiated in my previous class. Now, what happened is they differentiate into a lipoblast. Lipoblast means lipid forming cells. So lipolipid, so this is your functional unit of the cells. Now what happened is the lipoblast, they mature and they form either into multilocular, so these are multiple adipocytes that are present, or your unilocular adipocytes. So this will become your white adipose tissue, and this will become your brown tissue. No, there is no lipoclast. I haven't much known about it. Otherwise, fat, nobody will be obese then. So it will have lipoblast, and then lipoblast is divided into two forms: multilocular adipocytes and unilocular adipocytes. This is the photomicrograph. Now this is your white adipose tissue. So see this, these all hollow structures that you are seeing, these are your adipose tissue. Now, why they are looking hollow? This question should come, why there is no lipid that you can see or fat cells that you can see inside the cells? That is why, because they are microscopic structures. So what happened is, in order to prepare these cells to see it under the microscope, I have to go through many process. I do not want to go into the detail, or else it will be very confusing for you. But just for you to know that when they are going through the process, that process causes release of the fat, of the lipids. So when the lipid is released, they appear as empty. So that is why they are looking like this. So they are basically, see, they are unilocular. They do not have many locules. By locule means, see, this is one locule. So they are unilocular, and can you see the small nucleus that are present over there? So this is your single pycnotic nucleus. It is also referred to as signet ring cell. Okay, this cell, signet ring cell. So this is basically your a unilocular adipocyte, appears in standard microscope preparation as thin ring of cytoplasm surrounded the empty vacuole. So this is basically your very thin rim of cytoplasm that is surrounding your empty vacuole. Okay, sometimes referred to as signet single. Now, how does the eccentric and flattened nuclei? So this is your eccentric. Eccentric means at one end, it is pushed to one end, they are not in the center, eccentric. And your flattened, can you see the nucleus? This is normally, see, the nucleus is round, but in here you can see the nucleus is flattened. Okay. Brown adipose tissue, the color of brown adipose tissue or brown fat is due to both the numerous mitochondria scattered throughout the adipocytes and large number of blood capillaries. So it is basically because of the numerous mitochondria. What in mitochondria? It does contain mitochondria. So there are many mitochondria. That is why it does appears like that. And there is large number of blood capillaries. Adipocytes of brown fat contains many small lipid inclusions. So there are many small lipid inclusions and therefore called as multilocular, despite of your white cells which are unilocular. So what are the function? Same as for the white cells, they have the heat production. So the main function of multilocular adipose cells is to produce heat by non-shivering thermogenesis. Unlike white fat, liberated fatty acids of multilocular adipocytes are quickly metabolized with a consequent increase in oxygen consumption and heat production. So they metabolize very fast, unlike of your white adipose tissue, thus elevating the temperature of the tissue and warming the blood passing through it. The histogenesis is same, so you do not have to go through it again. So now this is your micrographic picture. Can you see? See, this is one nucleus, but these all are your multilocular droplets of your lipid cells. Okay, and here you can see that the cells are not clear. Here the cells are looking more dirty. And why? Because there are numerous mitochondria that are present, and there are numerous multilocular lipid are present. So these all are like that, and because of the presence of the blood and the capillaries, they are all imparting it as brown in color. Okay, so basically the cells are smaller, polygonal than that of white tissue, but their cytoplasm contains a great number of lipid droplets. All this that you are seeing inside are basically your lipid droplets. So now this is the difference between your brown versus white tissue. Localization, it is present subcutaneous, intra-abdominal, epicardial, gonadal. Whereas brown is present in your interscapular. Interscapular is near your shoulder muscles. Okay. Paravertebral, perirenal, cervical, supraclavicular. Clavicle is basically your collarbone. Okay. Morphology, it is spherical. They are elliptical and smaller than wide. Okay, now cell composition, it consists single lipid droplet, it has multiple lipid droplet. Few mitochondria, it has large number of mitochondria. Flattened peripheral nucleus, it has oval central nucleus. Then little endoplasmic reticulum, it doesn't have any information about endoplasmic reticulum. Function, storing energy. And what are the function of brown is expending energy and heat production by to non-shivering thermogenesis. Now see, you should be asking that what is non-shivering thermogenesis? Non-shivering thermogenesis is again a regulatory mechanism, a non-regulatory mechanism, sorry, in which your body doesn't have to feel cold. That is why this is called as non-shivering. You do not have to feel cold in order to send the signal to the cells that okay, warm my body. So they usually warm your body and the blood that is passing through it by a non-shivering mechanism. Okay. Now, what is the storage and mobilization of lipid? Where do the lipids are stored? How do they mobilize? How do they move? So the lipids stored in adipose cells are chiefly triglycerides. So you have to remember that. So the lipid that is stored in the adipose tissues are what? They are basically triglycerides. Okay. Basically, they are esters of your fatty acids and glycerol. Now, triglycerides stored by these cells originate in dietary fats brought to adipocytes as circulating chylomicrons. By that it means whatever we are eating, they break down into triglycerides and they are brought into adipose tissue through circulation by chylomicrons. So you have to remember that. The triglycerides synthesized in the liver and transported to adipose tissue. So this is how they come to the adipose tissue. That is why some of us have a thin structure, some of us have a broader structure based on one's dietary habit. Some are some are having more number of adipose tissues, some are having less number of adipose tissues. So what does it when we eat? That food breaks down and they turn into triglycerides. These triglycerides will synthesized in the liver. They are transported through circulation by chylomicrons to the adipose tissue in the form of BLDL. Okay. So this is what I have already explained you. You just have to go through with it. You just do not have to worry about it too much. You just have to remember like what is triglyceride, what are and how in what form they are stored as a triglyceride. Those things you need to remember. So this is all about today's lecture, guys. I'm stopping the